I'm Bruce Leuchter, and I am the CEO of Neurvati Neurosciences and GRIN Therapeutics. We can get into more detail on the distinction between the two. A neurologist and a psychiatrist by training. I've practiced neuropsychopharmacology for a number of years and have spent a number of years on Wall Street and in operating roles. GRIN Therapeutics is our first project within Neurvati Neurosciences. Neurvati Neurosciences is an operating company that works on projects in what we call asset companies or subsidiary companies, affiliates. And GRIN is our first project. It was brought into Neurvati at the same time Neurvati was formed in late 2021, September 2021. So we're here at our 3-year anniversary. And the nature of the transaction is consistent with what we typically look for in projects that we consider. So in GRIN's case, we had a small molecule therapeutic that had been in clinical trials in different indications, and so provided us with a pretty good amount of patient safety database, understanding of the molecule, understanding of the dosing protocol, PK profile, safety tolerability, the sorts of things that matter quite a bit to us as we assess opportunities. And GRIN Therapeutics targets GRIN-related neurodevelopmental disorder, which is a genetically defined developmental epileptic encephalopathy. And when we think about human genetics as it relates to our business, it becomes a pretty high priority on the list of priorities. So as we looked at the package, the molecule itself, and all the data that it had produced over a number of years, and combined that with a genetically defined disease, it became clear to us that pursuing GRIN-related neurodevelopmental disorder was a good decision. So we pushed off on that effort in September of 2021, and now find ourselves on the other side of proof of concept data, which is the reason we're speaking today in that disorder, which has no approved therapies. Patients with this disorder are treated empirically with antiseizure medicines and other medicines that help to control some of the non-seizure symptoms, neurobehavioral symptoms in particular. And given the robustness of the anti-seizure effect that we're seeing, as per our press release, a median of 86% seizure frequency decrease on top of background anti-seizure medicine therapy, we feel pretty encouraged and privileged to be able to deliver these data to the broader community, including the patients and advocates with whom we collaborate, hand in glove, and for whom the unmet need is profound. So very happy to be speaking with you today and talking about our progress. We think about the broader class of the syndromes that we target, GRIN being a part of that class of developmental epileptic encephalopathies. And that's a fancy term for progressive disease of the brain that leads to all sorts of difficulties. Seizures are obviously easier to see, easier to count, easier to assess than some of the other symptoms these children experience. But it is not the only symptom that we focus on. And part of the excitement surrounding our efforts in GRIN neurodevelopmental disorder is that the biological rationale associated with our compound and with this indication is a very, very good fit, not just for seizure, but for everything else. So the GRIN genes plural, encode for the NMDA receptor. The NMDA receptor has different subtypes. There's the GRIN1 subtype receptor, the GRIN2B subtype receptor, GRIN2A subtype receptor. And when you have a gain of function variant, so a mutation in the gene that leads to a gain of function phenomenon, you've got hyperactive receptors. And that hyperactivity interferes with all sorts of neurological and psychiatric symptoms. And our efforts around targeting in what I refer to as a rifle shot, as opposed to a buckshot, but targeting the underlying etiology of the disease process, in this case, a hyperactive NR2B receptor, it makes good sense that if we go and modulate that receptor, we could have impact not just on seizure, but on the broader syndrome. And if you look to your right and to your left in developmental epileptic encephalopathies, what you find is a substantial amount of innovation and progress made over the course of the last 5 to 10 years. A number of different approvals of drugs that treat DEEs, And almost all of them are broad spectrum antiepileptics, very good drugs that treat epilepsy, but don't necessarily treat the underlying cause of the disease. And that's what we refer to as the rifle shot for Radiprodil in GRIN-related neurodevelopmental disorder. So to take nothing away from the successes, the material successes of other compounds that have come through development successfully, we consider those programs to be broad spectrum antiepileptics. And in our case, we're focused on the specific cause of the underlying disease process, which is hyperactive NR2B receptors in gain of function patients. And by targeting those receptors, our hope for patients and for families is that we can deliver not only on seizure frequency decrease, but also on non-seizure endpoints, which is, of course, something we'll aim to prove in our phase 3 study. Gene sequencing, genetic testing is how it's diagnosed formally, where you can actually see the abnormality in the sequencing work that you do. But it presents very similarly to how other DEEs present. So kids who are not meeting developmental milestones, the standard progression of development over infancy and childhood, and they're not meeting those milestones, and they present to the pediatrician and the pediatric neurologist as a product of that. They don't have to have seizures to present in that context, but many do. And when they have seizures and neurodevelopmental delay, and they present to the doctor, what's become much more commonplace over the course of the last 10 years or so, certainly over the last 5, is to sequence these patients and to try to understand what their DNA looks like to be able to more properly diagnose these patients. Because in many cases, historically, before gene sequencing was a common tool in the toolbox, patients were diagnosed with autism spectrum disorder and other more grab bag diagnoses in lieu of being able to be more precise. And the good news is that now when patients present with neurodevelopmental delay and or seizure, they're plugged into the system. And that system includes a workup, a genetic workup, which can yield the definitive diagnosis based on the variants that they have born out of that testing. But GRIN-related neurodevelopmental disorder is an even newer diagnosis than many of the DEEs that have been targeted by drug developers over the course of the last 10 years. So this disease was only named in 2014, and the variant was only characterized in 2010. And as far as diseases go, that's a pretty young disease. So we work with the patient advocacy organizations and the leading principal investigators and key opinion leaders who are familiar with the disease, either by way of a research interest that's given way to significant work, or because clinically in some of these centers of excellences, academic medical centers, you've got pediatric epileptologists who are now able to characterize these diseases more precisely, and as such, they take interest in some of these rare DEEs. That's true both in Europe and the US, and frankly around the world. So while we know that any new disease requires a significant amount of legwork on disease awareness and relationship management with advocates who play a key role, and while this is a relatively new disease, we've seen a lot of traction, and we hope that the developments over the course of the last few days with our data will only increase that traction amongst the community and with patients who and caregivers who want to avail themselves of enrollment in the clinical study. So the study was a phase 1b study. We called it the Honeycomb study. And the primary endpoint of the study was to assess safety and tolerability in this patient population. Whenever you're studying a new patient population, you can never be sure of the safety and tolerability, even if you have hundreds of patients of safety data from other indications. So we ran this study primarily for safety and tolerability, with key secondary endpoints being efficacy. And the way that we assessed efficacy was to look at what we call countable motor seizures. And these are seizures that parents and caregivers can witness and document, which is exactly what they do in seizure diaries. And so we began by observing these patients over a 28-day period to try to get an accurate count of what their baseline is. How many seizures per week do they have? And do they meet the threshold for the cohort in our study that we call the seizure cohort? And in that cohort, there were the requirement was that patients had at least one seizure a week or for a month, so that when they're enrolled into the seizure cohort, we can assess the seizure frequency over the maintenance period of the study and then in an open label portion of the study. But there was also another cohort, and that cohort was the non-seizure cohort. They did not qualify as having 4 seizures a month or 1 a week. And those patients were still of significant interest to us because a number of symptoms that these patients suffer from are not a seizure in nature. They're neurobehavioral in nature. Patients have sleep cycle dysfunction. They've got impulsive behavior, affective dysregulation, which is not surprising given that we're talking about NR2B biology. And typically when you think about NR2B biology, you think about neuropsychiatric conditions. Many of our colleagues in the drug development world focused on this mechanism are focused on it for neuropsychiatry. So for us, the second cohort, the non-seizure cohort, was really important. And as luck would have it, within these 2 cohorts, 15 patients worth of data that met the criteria for the analysis demonstrated on the seizure side a mean seizure frequency reduction of 86%. That includes, patients who were on background therapy, so roughly 2 to 3 different anti-seizure medicines, and we counted their baseline seizure frequency on those treatments, and then came into treatment with Radiprodil, and those patients experienced an 86% seizure frequency decrease, which is remarkable, particularly given how severe these patients were with their morbidity in the disease process. We hear from the principal investigators that these patients were quite severe, and so to realize that 86% decrease was really encouraging. And then on the non-seizure front, we also saw directional improvement in patients. It's always difficult in an open label study with a small number of patients to assess, a neurobehavioral outcome. We did it across a number of different scales, the CGI caregiver, CGI clinician, the ABCC. These are rater-based scales that look at non-seizure endpoints in developmental epileptic encephalopathy studies and other studies. And what we saw was a directional correctness in those data. We will, at a subsequent date, dig into those data and talk more about them. At this point, with the top-line data release, we've indicated that these patients were assessed both by caregivers and by clinicians as improving in the study across these different parameters. But to win, I was going to say to win, this is clearly a win for patients, but to be able to perform in the way I just described, was really gratifying to see, particularly given how close we've become to the patient community and what parents and caregivers and patients, of course, contend with. So to be able to deliver these data was really encouraging. So we are developing Radiprodil, which is an NR2B negative allosteric modulator. There's a difference, important difference on how you modulate that NR2B receptor. There are some molecules that shut it all down. Those are called antagonists. And then there are negative allosteric modulators that negatively modulate the receptor, but don't outright shut it down. And that difference is key in terms of the therapeutic benefit that one can have in patients. And it differs from disease to disease, but in our case, in GRIN-related neurodevelopmental disorder, given that the GRIN gene encodes for the NR2B receptor, and in patients who have gain of function mutations, they have hyperactive NR2B receptors. So our negative allosteric modulator levels that fulcrum out, if you will. And that's the biological thesis in GRIN-related disorders. But the question we asked ourselves is, where else is NR2B biology implicated? What other pediatric epilepsies or neurodevelopmental disorders also implicate NR2B biology? And as we did the work on FCD and TSC, tuberous sclerosis complex and focal cortical dysplasia, there's some very interesting work, ex vivo studies primarily, where patients who've gone to surgery, which unfortunately happens to TSC and FCD patients when they become treatment resistant or prove to be treatment resistant to medicine, sometimes they go to surgery and neurosurgeons will surgically resect the parts of the brain that are thought to be implicated in promoting the seizure. And when you look at those tissues outside of the body, ex vivo, and you study them with NR2B antagonists or negative allosteric modulators, you see an effect. And the reason why that's not surprising is because this underdeveloped brain, these lesions, you can see them on MRI scan in TSC and FCD patients. You can actually see what they call hyperintense lesions. And those lesions are underdeveloped brain. And underdeveloped brain is known to have an increase in expression of NR2B receptors. So the biological thesis, while it is for genetically defined GRIN-related neurodevelopmental disorder, also applies to TSC and FCD in theory, because of the hyperexpression of NR2B in these lesions, which is the basis upon which we took Radiprodil into this additional study in TSC and FCD. So with these data, we now feel like we are in a position to have constructive dialogue with FDA, which is the next step in the process for us as we try to gain alignment around a single pivotal study for this disorder. Given that it's a rare, ultra-rare disease, there is a lot of constructiveness that companies like ours have with FDA, with regulators worldwide, frankly. And so our job now coming out of these data is to engage in dialogue with those regulators and to gain alignment around a phase 3 trial design. And then thereafter, kick off that study and begin enrolling patients in what we hope to be the final study required for approval. Because you mentioned TSC Alliance, and we, in parallel with the work we are doing in GRIN-related disorder, we have also kicked off a basket study in TSC and FCD. And the reasoning behind that is because NR2B, the NR2B biological thesis, in our view, applies to those disorders as well. So with any luck, we'll be able to reconvene in the future and talk about that study. But important to note that we believe in Radiprodil and we believe in its potential, and we believe in that potential beyond just GRIN-related neurodevelopmental disorder. And as we move into TSC and FCD, we hope to realize success there as well.